So maybe for the one person who still did not understood exactly how deep neural network works, I will still do the introduction, and I guess it will be a bit different from what you heard this morning. So the basic idea is very simple, is that what a deep learning model does is just take numbers as input and give out other numbers. That's a very famous example where you would give the algorithm an image with eventually a number written on the image, and it will give you a number which corresponds to the number which is written. Actually, it doesn't work like this exactly, but rather like that. So the numbers that you will, sorry, I just have a problem with that. The numbers that you will input here are the intensities of the pixels. And the number that it will output is not 8, but that's a classification problem. So there is a class 1, class 2, and so on and so forth. So what it will output is a vector with hopefully zeros for the number 0, 1, 2, 3, 4, 5, 6, and on 1 for the number 8. So how does this work? So maybe you've seen this slide or a similar slide before, but basically the first thing to understand is how a neuron works. So as you know, a neuron works by sensing here electric inputs, which are denoted by X here. And then it attributes a weight to each of these inputs, which are the W's here. Then it makes the summation of these weighted inputs, and then it will give an output if the sum reaches a threshold. There is a nonlinearity here, which can be a sigmoid activation function or else. So this is very simple, and then when you do deep learning, you add a lot, you make a pile of these neurons. So this is now a real network for the real task, which I just described in the first slide, which is here I have a 28 by 28 pixel image, so 784 pixels. Each of these pixels will be attributed one value, the corresponding intensity in gray levels of the image, and the output will consist of 10 numbers, each one corresponding to one digit. And in this, for this input, what I want to have is that the output of this whole summation, so here you can have negative numbers as well, all this summation plus this activation function will give me, for 8s only, zeros everywhere except for this neuron. So how do I achieve that? Well, you've understood that the trick is to find the good weights. So here each line corresponds to one weight. So I have lots of parameters here in this network. These are the weights. And I need to find the good weights. I don't know what are the good weights. There are many solutions, actually. And an easy way to do that is to start with random initialization of all the weights, which will give me a random output. And then training, that is optimize the weights. So you change each weight a little bit, and you see whether this brings you closer or further away, from the expected output, which is in this case 0, 0, 1, just for the 8 digit. And then you do this over and over and over and over. And in the end, you have a network which is able to recognize digits. So just a short parenthesis, you may have heard of convolutional neural networks, which are widely used for images. And these are the networks that we are also going to use for sequences. So just to mention that these networks, they are essentially like these ones. These are called fully connected because all the neurons on one layer are connected to all the neurons in the upper layer. Convolution layers, they are the same, but much simpler. So instead of having all the connection, you just keep the connection between a certain number, here 4 of the first layer with one neuron of the upper layer. And the thing is that here, the same color has the same weights. So instead of n times n here parameters, I have here only 4 parameters for this layer. So many less parameters to train, so you can put a lot of these convolutions. And what is nice about this convolution layer is that they have spatial invariance. So basically here what you do, you recognize you have a convolution of these motifs here through the layer. So this is very well adapted from problems like images where you have translational invariance. And in the case of DNA sequences as well, you have translational invariance. What you want to find is DNA motifs. So sequence of letters that you will recognize on the sequence. And these convolutional neural networks, they are especially good at doing that. So having made this short parenthesis of convolutional neural network, let me finish this introduction by telling you what you already know is that to train the network, you need a lot of examples. So that's why some people, they have, so this is, by the way, the Yann Lequin that Cedric was talking about. So maybe the first application of deep learning has been digit recognition, and he has, to train the network, managed to make this hand annotation database, which is called NINST, which is very famous. So you need a lot of examples. I have to come back.
Yeah, just give one click so it selects. And now you can...
And this is the reason why he developed this Handdigit recognition algorithm for check banks to recognize what is the number which is written in the rectangle, because when you get many of these checks, then you want a machine to read it and to credit the account. So about learning with computers, this is a very old-fashioned topic. So I just found this book somewhere in the lab, which is called Computer-Oriented Learning Process, and which dates back to 1974. And in the first page, in the introduction, we can say that there was a NATO Advanced Study Institute conference on the topic of learning, which starts by 20 years ago. So in 1945, the topic was quite fashionable among the true believers of machine intelligence. Unfortunately, over-optimistic prediction did cast a general discredit on the subject, which among many kept a flavor of poor science fiction. So the idea is old, and it's a long time that people have said, okay, it doesn't work. So what happened now that it works so well? 3 things, combination of 3 important things. You can't you would not have the rise of deep learning now if you don't have those 3 things. The first one is GPUs, graphical processor units, which were developed for video games and which are very good at making parallel computation. The type of computation exactly that you need to train the networks. The second thing is storage. You want big databases with a lot of examples. So you need a place to store all these examples at one location where you are going to train your network. And the second thing you need is the internet, because all these images, they can come from many different locations, and you don't want to bring them with horses, you want to bring them with wires, and that's what the internet is doing, putting all the data at the same place so you can train your network. So that's why now you hear a lot about deep learning. So these are very famous example that you know, that you've seen, but the famous AlphaGo system, who is now beating humans at Go, which was previously thought to be impossible, and which is based on adversarial learning. The idea is that it can recreate virtually an infinity of games just by playing against itself and getting better all the time, and even better than all the Go masters. You have the recommendation system of the GAFAs, so they have made all their reputation mostly based on algorithms derived from deep learning, where they can tell you what you need, when you need it, and even if you don't need it, you will understand that you need it. They can be used for automatic driving. So you have seen in the lecture of Cedric that networks, once they are trained, they are very fast at taking decisions, and this is what you need. You want to make real-time decisions when you drive a car. So only from the pixel intensities of the images which are taken from a camera, which is showing you what is in front, in the rear, and on the side, you can take the decision to accelerate, to brake, or to turn. And in biology, this is also a famous example of what deep learning can do. So this is this CASP, critical assessment of structure of proteins. It's a contest which is held regularly. And basically, this contest is, can you tell me the structure of a protein if I give you only this sequence? What people do is that, okay, we take 5 proteins, we make the experiment, we get the structure, we don't give the structure to anyone. You give me all your prediction, you tell me what is your method, and then I will tell you who was closest to the reality. And the performances of the algorithms were pretty steady in the past 10 years. And then there was Google and AlphaFold, and this is what happened when they started using deep learning to make this prediction, which is complete change of performance here. So the question now of the talk is how good it can be to interpret DNA sequence. What can we learn from applying deep learning to this object, which is a sequence of A, T, C, and Gs? So as you know a lot about deep learning, I'm not sure you know so much about genomics, so there is also a short introduction about genomics here. So this is the DNA double helix. And the DNA double helix in chromosomes, in our chromosomes, is wrapped around proteic spools, which are called nucleosomes. So this is true only in eukaryotes, that is organisms which have a cell with nuclear membrane, a nucleus with a membrane. And I will focus on eukaryotes here. So eukaryotes, they have nucleosomes. And then these nucleosomes, they are somehow folded in higher order structures up to the way to the chromosomes. So what is very nice about these nucleosomes is that they have 2 roles. And the first role is a structural role. The second role is a gene regulation role. So the structural role is to soften the DNA molecule. The DNA molecule is very stiff. So the Young modulus of a DNA molecule is the same as if you put it on scale as a raw spaghetti. It's very stiff, raw spaghetti. So if you want to fold a long genome into a small nucleus, then you need to soften it a lot. And basically, that's why this does. You see it's very, very bent around these nucleosomes. And also it's the bead on a string array of nucleosomes is very flexible compared to a single DNA molecule. The persistence length is reduced by a factor 10. So this allows you one great thing, is to have much bigger genomes. And this is what you see here in this graph. So in this graph, what I represented is the number of genes, so being the number of proteins that are encoded within the genome, to put it simple, with respect to the genome size. And this is log-log scale. You see a very linear relationship between the number of genes and the genome size for viruses and prokaryotes. Prokaryotes, they don't have a nucleus, they don't have nucleosomes. So basically what it means is that for these organisms, genomes, they are just a collection of genes put side by side. And basically that's what your genomes. What is your genome? For eukaryotes, which have these nucleosomes, then there is a completely broken here relationship and an explosion of genome size with approximately a constant gene number. So what does this genome size explosion allows you to do, which you could not do when you were a prokaryote, is to make cell differentiation. That is, with the same genome, you are going now to make differential regulations and to have different cell types, blood cell types, liver cell types, muscles, neurons, whatever cell types you want, and you are able to make multicellular organisms that will undergo development. So that's why these nucleosomes, I say they also have a gene regulation role, and I'm going to explain it a bit further in the second slide. So I told you about unicellular eukaryotes. For instance, yeast is a unicellular eukaryote, so it has nucleosomes, but it still has a very short genome size. Basically, it has it maybe derives from a multicellular organism, so it's now unicellular. And the genome in yeast looks like this. In orange, you have the genes. Then you have the promoters of the genes, that are the regulatory regions of the gene, which will tell you if the gene is expressed or not. And they are side by side here, very small genome. If you go now to humans, you have huge regions of DNA between the regulatory elements and the genes. And in fact, the genes in the human genome, they only encompass 2% of the genome. 98% of the genome is not coding for any protein, but mainly we don't know what it is. These regulatory elements, which have been identified so far, they account for a very small portion of the genome as well, 0.5%. So what about the nucleosomes? Well, the nucleosomes, they have a very clear relationship with the regulatory elements. So these are the small sequences here, which are usually 100 base pair, which are key to the regulation of genes. And in the yeast here, the nucleosomes, they are only found closely packed on the gene bodies, but not on the regulatory elements. And in human, it's the same. The regulatory elements, they are devoid of nucleosomes. So basically, you can think of nucleosome like spools, which store the DNA, which is not used for regulation, but the DNA which is used, which is bound by transcription factor, is free of nucleosome. So the nucleosome, they are like a reading frame of your genome. They will tell you where are the sequences which are functional. And in that sense, they have this gene regulation role. Or at least they are a print of the sequences that are useful for gene regulation. So now coming back to the sequence, well, when you have a sequence like this, you don't know if am I in a gene, am I in a regulatory region, where am I, what is this sequence doing, you don't know, you have no clue. So one thing that would be good is to have a tool that takes a window here and tell me, is it a gene or not? Or tell me, is there a nucleosome in this sequence or not? Because if you can do that, then you have the reading frame of your genome. You know where are the regulatory sequences and you know where are the genes. And that's what we are going now to see how we can achieve that with deep learning. So the first part will be on where are the genes, and the second part will be on where are the nucleosomes. And there will be maybe a short part on how to use now deep learning to make new sequences, synthetic sequences, with defined properties. So I'm going to simplify a bit the problem for the first part. I'm not going to look at where are all the genes, but just I'm going to focus on the gene start site, so the beginning of the gene. And the gene start site, which is here given by this dotted line in the box, they are called also promoters, and they are the region where the machinery for transcription of RNA will come and start to transcribe the gene. So we really want to locate these regulatory elements because they tell you where to start. We don't focus about where to end for now. So the question will be that if I give you, let's say, a 200 base pair sequence, which is centered on gene start sites, can you make a machine learning algorithm, can you make a deep learning model which will tell me if this sequence is a gene start site or not? Okay. So to do that, we need to train the model, and for that we have to, what we do is that we take all the known gene start sites in the human genome. To get those gene start sites, you need a lot of effort. So we know them very well in human, we know them quite well in mouse, we know them in some organisms. For most organisms, we don't know them because it's a lot of experiments, it's a lot of money, it's a lot of work. But for human, well, we spent a lot of work. So now we know that there are approximately 30,000 gene start sites. We can collect the 200 base pair sequence which correspond to these start sites. And then we can take, okay, let's take some negative examples. So I take sequences somewhere at random, same amount of sequence which are not gene start sites. Then, so this is my sequence which I extract, ETEACGD. I take 200 base pair, and then I have 4 letters. So I transform my sequence in an image 4 by 200, and I put a 1 if I have a T on the 4th line, and I put a 1 on the 1st line if I have an A, and so on and so forth. So I transform my sequence into an image, basically. Very simple image, which is only 4 by 200 pixels and which has only zeros and ones. But still, it's a very nice grayscale image. Then I will put my image into a deep neural network, pretty similar to the one I showed you before, where we use convolutional neural networks. And then, as an output, I would like to know if it's a GSS. So if it's yes, I would output a 1. If it's a no, I would output a 0. And then I can do the training. And once it's trained, whoop, yes?
Why is it advantageous to do that instead of just looking at the sequence? Just keeping the letters?
Well, because you want to enter numbers, basically.
Why? It could be 4 numbers?
Yeah, you can do 1, 2, 3, 4.
Yeah.
But this is advantageous because then you have a symmetry between all the letters. So if you take 1, 2, 3, 4, then which one would be 4, which one would be 1, which one would be 2? Then you are going to multiply by numbers, and it's not exactly the same. This process, when not in coding, it's really good for classification because all the class, they have exactly the same role in this embedding. So this would be your training set. So we are going to take all the gene start sites present on all chromosomes except the 21. We leave it to make the test set because then we want to test our network on some sequences he has never seen. That's the where we are going to do the prediction. So here is the result. Now what we do is that we take the sequence of chromosome 21, which the network has not seen, and for every 200 base pair, with a sliding window, we compute with the network the probability to be a start site. So we get a number between 0 and 1, and this will give us, so this is a very short window on the chromosome 21, which is our prediction set, and this is the answer. So you get this noisy signal. And you notice that, so these are now the real annotations. I made a simplified description here of these annotations. So these lines, they are the real gene start sites. And you see that on gene start sites, you have a peak most of the time of your signal. But your signal is very noisy. The reason why it's very noisy is that the real sequence has an imbalanced number of 1 and zeros. It has much more zeros, in fact. Most of the sequences are not gene start size. So if you want to restore this problem, that's our contribution in this work, is to say that you should not take only 30,000, that is the same number of non-gene start size sequence, but you have to take much more. So that's what we did with taking the same network, but training it with many more negative examples. Here, 3 million negative examples, and then you get a network which has a very clear signal and which can spot the gene start site precisely. Then you can make a picture which is an average of these signals over the gene start site position here at 0, and then you have a nice peak of the prediction around 0, which is much sharper. That was the purpose of this paper in the case where you take 100 times more negative example than in the case that you take a balanced number of examples. The reason why we did that is that in the field of machine learning, usually people like to train with balanced data sets. And in genomics, the machine learning people who started to do that, they did this with a balanced data set, but they did not get so good results. That's why we did this. Yeah.
It's not so intuitive to me because here it seems that you further increase the imbalance.
Yes, yes, yes.
And that's quite counterintuitive because in the field you would try to, of deep learning, you would try to reduce the imbalance.
Yes, yes.
So can you explain that again?
Well, the real data set is very imbalanced. So we are learning on something which looks more like the real data set. There is a trick here, is that we don't do the learning exactly in the same way that it is done in the case of a balanced data set, but we attribute weights to the different examples. So we would give more weights to the positive examples. So the network really has to learn the positive. If you don't do that, you will get zeros everywhere because, well, one time on 1,000 it will be a 1. So if the network always outputs zeros, it will be happy and will always tell you 0 the answer.
So you use a weighted layer, classification layer.
Yeah. But why? Yeah. Maybe you're asking why. Do you want to predict the gene start site on chromosome 21 when you know already from the experiment the start site on chromosome 21? Because you cannot measure only on chromosome 1. When you do the experiment, you measure the gene start site on all chromosomes. So why? The reason why, one reason why, is annotation transfer. As I told you, the human genome is very well annotated. We know a lot of genes on the human genome, probably all the genes on the human genomes. This is not true for most of the genomes. Actually, for the mouse, it's not so bad, because the mouse is a medical model, and we have very well annotated the mouse as well. But the idea is that, okay, can we train now a network on a very well annotated genome, let's say the human genome, and then predict what will be the annotation on the mouse genome? So this is what we try to do. So this is the results which I just showed you, trained on human and predicted on human. And now this is the result if you train on human and we predict on mice. And you see that, well, it works approximately the same, almost as good. So we can predict gene start site in mouse as well with the network. And this is the other way around. Now we train on mouse and we predict in human and still it works. So we say, okay, that's a good thing. Now maybe we can annotate more genomes.
Naive question maybe. Is there any gene start site on chromosome 21 that does not appear in the other chromosomes? Or
Yes. All gene start site on chromosome 21 does not appear in other.
They are specific to chromosomes.
Yeah, yeah.
Okay, I see.
Yeah. So for this case, we did not use, in fact, the chromosome 21 because they're different in mouse and human. So we use the chromosome X because the chromosome X in mice and human is the same. So we had to use another one. Just a detail. So then we say, okay, it works with mouse. Okay, what about chicken? So chicken is the best annotated genome in. Remember that we need to take annotated genomes to see if our algorithm is working or not. So now we tried the chicken because the chicken is the best avian annotated genome. And I don't show you the results here, but we show where it works. I can train on chicken and predict on mice. I can train on mice. So, okay, this works. And what was interesting, maybe I should have put the result because there was something interesting here, is that if you train a network on chicken and predict on chicken, so on a different chromosome, for instance, chromosome, well, never mind, a chromosome where you didn't see in your test set, it doesn't work as well as if you train the network trained on human. So it works better if you use the network trained on human. And the reason why is that the human genome is already much better annotated than the chicken genome. So already the network that you train on human can tell you something about chicken that you did not know. And then we say, okay, now can we go to the fish? And no, we cannot go to the fish. It doesn't work at all. So there is something in evolution, that something happened in evolution, and actually we know already a bit what it is. It has something to do with DNA methylation that makes a big difference between the way genes are regulated in fish and in these species here. And in fact, if you want to annotate a fish genome, then you have to train on fish genomes as well. So it's not universal so far. But maybe a prospect would be to now annotate with all annotation from all genomes known, and then make a kind of super network that we could use on any organism. But we are not there so far. So this is just to conclude on this part and to say that this is important to do that, because here you have the number of completed genomes. So more and more DNA sequencing is done, more and more techniques are available, more and more algorithms to have complete genomes as well, so that you see this exponential increase of number of genomes that is obtained every year with here an artifact. And you realize that to annotate all these genomes, well, it's simply impossible to do now with experiments. We will have to rely on computational methods. So now, still why? Okay. Second application is to assess the effect of mutations. Because now that, okay, let's say the start site, I know if I give you a 200 base pair sequence, if it's a start site or not. What happens if I just change one base pair in this start site? Will it remain a start site or will it not be a start site? Or inversely, if I have a sequence which is not a start site, can I change it to a start site just by changing one base pair? That's the type of question we can now address with the network. But we will not do that. Instead, we will go to nucleosomes again. So nucleosomes, as I told you, are the first level of genome organization in eukaryotes. So these are these pools. And there is something I did not tell you about these pools, is that they are regularly spaced along the DNA molecules, except when there is a regulatory element, a green element, you remember. But that the distance here between nucleosomes, it can vary a lot. It can vary between organisms. It can even vary within an organism. It can vary in different cell types. But usually it's between 20 and 90 base pairs, where here around you have 147 base pairs, always. So how to know where are nucleosomes on the genome? For this, there is an enzymatic way to know. You use MNAs here, macrococcal nuclease, which is an enzyme which eats DNA, but only if it's not wrapped onto a nucleosome. So only this DNA is eaten here, and you end up with mononucleosomes. Then you read the DNA sequence of all the DNA which is preserved, and then you map back these DNA reads on the genome. And what you end up with is a density of nucleosome along the genome. You see these nice peaks. The fact that you see peaks, in fact, means that when you do this on a population of millions of cells. The fact that you have peaks means something, is that nucleosomes occupy the same position in all the cells, approximately. Or at least there is a strong impact here of the DNA sequence. And then there is a question which is left, is how does a DNA knows to go here but not there? What makes it go always on the same sequence? And a way to answer this question would be to you already know what I was saying, but first I have to say that. So if I go back to my example of yeast, nucleosomes are in the same position, but in fact you already knew that. Yes?
So these histograms come from the same tissue, from which kind of cell?
So they come from here a yeast culture. So it's not a tissue, it's a population of yeast cells, unicellular. But it could be a tissue as well. Huh? Yeah. Yeah, if you would do it on different tissues, you would not have this clear pattern. So coming back to my yeast here, you remember these are the piece of the yeast genome here. These are the genes in blue. You remember what I told you, that the genes, they are side by side. They occupy almost all the genome. And here, between the genes, you have regulatory regions, which regulate the genes, which are here shown by these dashed lines. And here is an experimentally determined map of nucleosome position. And what you see is that, okay, nucleosomes, they are regularly positioned over the gene bodies, and then there is a drop of nucleosomes at the regulatory sites. Drop, drop, and so on and so forth, which is exactly what I showed you on the scheme before. So, but that's a real data. And now the question will be, can I train a deep neural network so that I give him the sequence, and he tells me what is the density of nucleosome on this sequence. So that's exactly the same of gene start sites, but instead of having a binary signal, 0, I'm not a gene start site, 1, I'm a gene start site, here I have a continuous signal. So I normalize this signal here between 0 and 1, and now I pair each sequence here with the value of the nucleosome signal in the middle of the sequence. And I train a network by giving him these sequences and trying to predict what is the signal here in the middle of the window. Exactly as in the gene start site case, I take a lot of chromosomes to train the model, and one chromosome I keep aside. Now I have 16 chromosomes because I'm in yeast, you remember, for the prediction. And this is what I get. So this is the same region which I showed you before with the genes here. This is the experimental signal. This is the prediction. And you see, okay, it's pretty good. Actually, you can plot the experimental value versus the prediction. You have a correlation of 0.68, which is really fine compared to better than methods which were developed before, which did not rely on deep learning. Okay, but that's something very classic. Okay, deep learning is better a little bit. You increase the correlation. Well, what is interesting is that we tried with different lengths of the sequence. And we start with 200 base pair, because basically one nucleosome is 200 base pair. So we would say, okay, 200 base pair should be sufficient to know where a nucleosome is. But not at all. In fact, we could get only good prediction if we increase the size up to 2,000 base pair, which is approximately the size of the genes here. And the reason why is that, in fact, what the network is doing is that it's spotting the regulatory regions. And then it's looking at what is the distance between the position where I stand and the next regulatory region, and then it's giving the amplitude of the signal based also on this distance plus some sequence feature. So you really need to know how far you are from a regulatory sequence to know how nucleosomes are phased.
Yeah. If I understand correctly, here you're using a convolutional neural network.
Yeah.
Have you tried using a recurrent neural network instead to see how if it gets better?
Yeah. It's exactly the same. Basically, we tried a lot of architectures, and we always get the same correlation. It's not, somehow it's in the data, it's not really in the architecture, which is a good sign that at least it's not a problem of an architecture, but yeah. So now that we have this network in hand, we can do what I was telling you that we were going to do in part 2, is to look for the effect of mutations. So here is the density of nucleosome over a 2 kb region here in blue, the wild type sequence. Now, here at point 0, I change the A here, and I replace it either by a C, a T, or a G. And then I have the profile over the mutated sequence. And then I make all the possible mutation. And then I see what the difference for each mutation. I look at the area here, the difference between the two signals. And I make all the possible mutations. And then for this location, I have a mutation score, which tells me how important is this precise nucleotide for nucleosome positioning. And then I can compute this mutation score for the whole genome. That's what we did. So for all the chromosomes, we computed the effect of every nucleotide to nucleosome positioning. And this is the signal that we got on a portion of the chromosome 16. So most of the time it's here between 1 and -1. So that's a Z score. And sometimes it peaks. And then if you look at where are these nucleosome depleted region positions, so these nucleosome depleted regions, they are the regions which are between the genes, the regulatory elements, if you remember, which are between the genes. You see that often the peaks here, they are found at this nucleosome depleted region, which is in fact expected. The interpretation is that the way nucleosomes are positioned on the yeast genome is that you have specific sites where you repel nucleosome, And then nucleosomes, they tend to pack against these sites from which they are repelled, against the border of these sites. So that's where the mutations have the maximum impact. And then you can look at what are the motifs which are implied in these changes. And for this, we take the distribution of the mutation score. So as you see here, most of the time it's between 0 and 1. So that's this big distribution here. And there is this fat tail here towards high mutasome score. And we took all the sequence here which were above 5, somewhere here. And then we just looked at the sequence which were here. And what we find is this. AAAA, TTTT, or CG, CG, CG. And in fact, actually, we found something which was already known, which is reassuring. We would also have liked to find something which was not known, but we did not find something which was not known. We found only known things, which is that these poly A and poly T's sites, they are very difficult to bend, and they never form on nucleosomes, so they tend to repel nucleosomes. We also found these CG sites, which can be bent easily, but these sites, they are known recruitment sites for an enzyme which is known to displace nucleosome as well. So that's why we recovered. The good thing is that, well, we say, oh, this is sufficient now to position nucleosomes on the yeast genome. You don't need an extra mechanism. Only these rules are sufficient to position nucleosomes as they are experimentally found. So I have time for the third part.
Can I ask you, because
maybe
between those start sites, you have still some modulation of this position.
Yeah.
Does it refer to this kind of motif AAATTT and so on?
no, but no. But yeah, you're right that we capture really fine details of the densities, which are not related to that. So maybe if we go to lower mutation score, the fact is that we only look for a single mutations effect. So these modulation, we don't know what causes them. Basically, it can be a combination of many motifs, and we don't know, basically.
Does your score somehow match with this positioning, intra-start site positioning of this nucleosome, or not?
You see, we did not investigate in too many details, so we were really happy with that because it was a lot of fun. At one point, you have to publish the paper, and then you have to think of something new. But basically, there are. It's not always that the high mutasome score coincides with this nucleosome depleted region. So these one, they would be most of the time this site. Of course, we look for enrichment. There are also some peaks which are not this sequence, and we don't know what they are, basically. But they are not statistically present a sufficient amount of time to come out from a statistical analysis. So we just didn't look at them. But we could be more analyze this with more careful. I mean, what we did is that we took this signal. Now it's done. We publish this, and we say, OK, now you can download it and look at whatever you want. So I can go for genome writing. 15 minutes? Okay, but that's 2 minutes. So the third reason, third possible application of this is that now maybe you can write genomes. So I'm not going to show you that we wrote a genome, but just a very small piece of genome. And here is what we wish to do. I told you that the distance between nucleosomes, it can vary in different organisms between 20 and 90 base pairs. Well, it happens that in the yeast, it's one of the smallest one that you can find on living organisms. It's 20 base pair. They are very close by. In the sea urchin, it's one of the longest. It's 90 base pair. It's much, much further away. And what we ask ourselves is that, OK, can we turn yeast chromatin into sea urchin chromatin? So can we design a sequence that we will insert in the yeast where we will have a spacing of nucleosome that would be of 90 base pairs instead of 20 base pairs. So we were, for this, focusing on a very simple way to do that, which is to create one sequence, which will be 237 base pair, and then that we will assemble in tandems, many of them, maybe 10 kb, and then insert into a yeast chromosome and measure the nucleosome density on it. So first of all, we said, okay, how would look like the nucleosome density on such a sequence of 237 base pair, which will be assembled in tandems? And what we started with is to take a random sequence of 237 base pair. And then we create a synthetic sequence where we just put this sequence side by side. We make a lot of them. And then we use our network to predict the density of nucleosome over this sequence. And what we got is basically nothing. That it's a 0.5 everywhere. We took a random sequence. Nucleosomes are not well positioned on this sequence at all. And then we said, OK, this is our target. Our target is one nucleosome positioned at the beginning of the sequence, then a big hole of 90 base pair, and then a second nucleosome. And now we want to change the sequence here so that this density predicted will reach this target. And what we do is a Kinetic Monte Carlo approach when we mutate all the base pair. So 237 base pairs, and we made all the possible mutation. That is, if we have A, we tried C, T, G. And then we attribute an energy to each change, which reflects how more likely it is to the target than the other sequences, than the other mutations. So then, now that we have an energy for all the sequence, we have a probability for all the sequence, and we pick one mutation at random, which is a priori more likely to go towards the target. And we do this for a few steps. 1, 2, 3, 4 steps. And already after 4 steps, we see, so we have done 4 mutations on this 237 base pair sequence. And we see that the prediction already starts to look like something which looks like the target. And after 20 mutations only, we have a density of nucleosomes which is very close to the target. So if you start from a random sequence, you just change less than 1 out of 10 nucleotides, and you already make a sequence which looks like that. After, you can continue, and you just generate many more sequences which will have the same nucleosome positioning pattern. And you can, in fact, generate really a lot of them and keep it like this going forever. Okay, I don't have the slide, but I can tell you. So some of them are obviously in these nucleosome depleted regions, and many of these mutations, they tend to make stretches of A's or of T's, and some of them are at the dyad of the nucleosome, so really the middle of the nucleosome, and these ones, they tend to break stretches of T's and of A's. There are also more complex things, but basically something which is quite, in fact, understandable based on the rules that I presented before. So now, experimental validation. So in collaboration with Jean-Baptiste Boulet, we synthesized these arrays of sequences, and we used 3 different cases. In one case, we made the sequence of 167 base pairs so that we have the same spacing than in yeast. We took 50 base pair, 197 to have the same spacing as in the rat liver, and we also took the extreme spacing of the sea urchin. So these are the targets. These were the targets I showed you before. These are the prediction for which we choose to synthesize the sequence. So it's not as good as the one I showed you before. But still, these are the experimental densities. And you see that in each case, we managed to position the nucleosome where we expect them to be. Just on a short note, we did that because before that, we used a sequence here, which was known to position nucleosomes in vitro, which has been obtained by selection experiment done in vitro. because we wanted to make a regular array of nucleosomes, and it was completely failing to position nucleosome in vivo. So we really had to do through this stuff to get what we wanted. So this is one very small example of starting to write genome, and just to mention that there is now a big effort in genome writing, and there is a consortium which is called Genome Project Write, which says that while the sequence of the human genome has been known now for 20 years, many mysteries in life received book remain to be solved. That's why one group of researchers, as well as ethicists and communicators, is proposing to move from possibly reading genomes to actively writing them. So this is something which is happening now in the next decade. So these are the people who did. So I only presented people work here which was done within my group. So mainly from Etienne Routier, which is having his defense very soon, a PhD student. And this is Jean-Baptiste Boulet, my experimental collaborator in the lab. And thank you for your attention.
So I have two questions. One is related to what you've just presented. Could you actually use your design technique to tune, for instance, the regulatory properties of genes, or change the strength of a promoter, or that kind of things, just by shifting nucleosomes or something?
Okay. So that's what we want to do now. So basically, here we engineered sequence which are regular arrays of nucleosomes. And one of the questions is there a length starting from which everything will be transcribed? So this is a part of in relation to the question you asked. Now about gene regulation, there has been big work by some American groups that used on top of deep learning, to train their model, they used a library of millions of random sequences for the regulatory region, and they looked with a rapporteur what was the expression of the gene based on the sequence. And based on that, they trained a deep learning model based on the results. And now, I would say we have really, you can type in your sequence and you will know what expression it will give.
Okay, so that has been done. Okay, so that was my first question. The second is a bit more unrelated. As an expert in 3D genome organization, did you, let's say, try, or are you willing to see whether you could develop something like AlphaFold for the 3D genome organization, and how? And
so I think it's much less interesting than in the case of AlphaFold, because in the case of AlphaFold, the function of proteins is really driven by the structure. In the case of genome, we know that the activity of the genome is somehow reflected by the shape. But it's very less well clear that the shape really impacts the function. So I don't think it's as important to do that for genomes. So I would not do that. But some other groups tried and partially succeeded in doing that. So if you're interested, there are, I think, 3 papers now that predict the 3D folding from the sequence.
Any other question? I have just a short one. I was wondering if your synthetic mutation on told you something new about nucleosome positioning that you didn't know. So you told us about rigidity and maybe binding energy or something. Did you learn anything?
No, not really. Not really. I think we are going to learn something new now that we are moving to do the same thing in mouse and humans. Because in mouse and humans, there is some, in yeast it's simple. I told you, you have nucleosome depleted regions and then stacks of nucleosomes and nucleosome depleted regions. And then there are physical models who just reproduce the positioning of nucleosome based on the fact that you have these repelling regions, you have steric interaction between the nucleosomes, so you have, in fact, a gas model of nucleosome which reproduce quite well the data. in human and mouse, the distance between regulatory elements can be tens of kbs, and still you have a regular positioning. So it means that there is something in the sequence that position nucleosomes even far away from these regulatory elements. So here, I guess there would be something like that, but in yeast, no.
Thank you. That's it. All right.
So you mentioned this problem of balance or imbalance of the training set. So basically, it comes down to having a prior information about the probability of one particular sequence to be a start site or not. So is there other ways to inject this prior into the training rather than just balancing or imbalancing the training set?
I think I don't understand the question.
So when you remove the balance between the training set,
yeah,
you are basically using a prior that start sites are rare compared to.
Okay, but that's
right?
fact. I mean that's.
Yes, but you give this prior to your network,
yeah,
by giving unbalanced examples. That's how you say
yeah,
the network that it should be rare.
Mm.
And the question is, do you have other methods, computational methods, which I mean would be some kind of Bayesian modulation of the output of the network or something similar, to inject this prior into your predictions?
No, we don't.
It's always just by. How did you decide this 1%, for example?
Okay. So we start with 1, and then we start increasing. So it's purely experimental, I would say. We start 2, and then we gave the whole genome. So all the sequences which are not start sites as negative examples. And what we found is that there is a learning curve. So first we have bad results and it's getting better, and then around 100 times more example, there is a plateau. So we suggested that we should use this. But this is valid for the human genome. I don't know how it extends to other genomes. So this is purely empirical.
But in this case, maybe you don't need to know in advance because you can just feed the whole genome. But then you said that the results were also weighted. And for this, I guess you need knowledge. And this I actually do.
Well, the weight is very simple. It's just the imbalance. The weights that you give. So if you have 100 more negative example, you just give weight 100 to the positive example. So depending on the data set. And then, yes, you can use the whole genome in any case, but it's just more time. So you don't want to lose time.
Okay. All right, so let's thank again the speakers.